Well, it's my pleasure to introduce our opening plenary speaker, Ed Buckler. Ed is a research geneticist at my previous home, the USDA Robert Holly Center for Agriculture and Health at Cornell, and also is a professor of genetics at Cornell University. And he's also the co-director of the Institute for Genomic Diversity at Cornell. So Ed is one of the premier plant biologists, and especially in the area of genetics. His work has been instrumental in dissecting agronomically important complex traits and linking them to specific DNA sequences, and taking advantage of natural diversity, and using this for enabling plant breeders to improve crops. He's developed in the plant side, in terms of association genetics, the statistical methods for association mapping and help solve some of the confounding problems we have in this area with population structure, and was the first to introduce mixed model statistical approaches to this in the plant side, and is currently still the most robust approach for this. He's used this for a number of successes in terms of identifying possibilities for crop improvement in terms of sweet corn, increasing beta-carotene levels. He went on to develop with colleagues the largest complex trait dissection platform of any species, the nested association mapping panel, which combines the strength of linkage and association mapping, and that has been used to help identify genes behind other important traits such as flowering time, plant architecture, disease resistance, etc. Now, he's like many accomplished scientists, he's received numerous accolades, and these are quite impressive, including the U.S. Presidential Early Career Award in Science and Engineering, and it's one of the ones he got when I was the director of the USDA lab. And I remember seeing a picture, Ed, with you in your suit. You were in a picture with President George Bush II. Yeah. So how we long for those days of leadership. He's also received the Arthur S. Fleming Award for outstanding federal employee, and there's only 3 chosen from the entire U.S. government. He was elected to the U.S. National Academy of Sciences in 2014, and recently was awarded the inaugural 2017 National Academy of Sciences Prize in Food and Agricultural Sciences. So without further ado, let's bring Ed up here, because I can't wait to hear what he has to say.
Well, first off, thank you, Leon, for the very generous introduction. And it's a real pleasure to come up here and visit you all. This is my first time to Saskatchewan, and so and I'm glad I'm coming during the summer. But it's a real pleasure to come visit you all and to really see some of the amazing science that's going on here. I'm going to, my day-to-day work is figuring out how complex traits work, and figuring out the genes that are at the basis of them, and using that knowledge to apply it to breeding. But today, because of the group, really interesting group of scientists we have at this meeting, I want to touch on 2 somewhat unrelated topics that I think are very important and hope will stimulate discussion throughout the rest of the meeting. So I want to highlight that you can't do any of this science without being supported by some really great institutions. And in my case, a lot of the work I'll be telling you about today has been supported by USDA ARS and by the National Science Foundation. And then I've had the incredible pleasure with collaborating with over 1,200 scientists from around the world in the last 20 years. And these are a few of the collaborating labs I'm working with right now that have allowed us to do some of our great research. But it's really been that focus of collaboration and working together that's allowed us to make some of the contributions we've had. So in terms of our lab mission, it's essentially 3 parts. The first is that a very basic science question of how do we understand the genetic basis of natural diversity? How does DNA get converted into something that shows up as a differential growth, differential yield out in the field? The second element is how do we breed crops for the world, take that knowledge, unify it together, and apply it in day-to-day breeding. And the third element that my group is focused on is figuring out approaches to reduce the environmental impact of agriculture. In today's talk, I'll actually start with that first one and then I'll tell you one vignette about how we're using some of the basic science side of things. So I have a background both in archaeology and evolution. And so when I think about plant breeding, I really always start thinking about where did we come from? Have we built upon the right set of decisions? What were the historical contingencies out there? And so I think we're now entering what I would call breeding 4.0. But it started back in breeding 1.0 when people 10,000 years ago were out looking at various species from around the world and deciding which ones to domesticate, which ones to select, and how do we integrate and put together that diversity. Then starting a little over 100 years ago, controlled trials really come into being, where quantitative genetics, experimental design, statistics, and important equipment like harvesters that can accurately measure the phenotypes out in the field really become the key elements of breeding 2.0. And breeding 3.0 started about 15 years ago with genomic selection and really effective marker-assisted breeding, where we're using genomic information to accelerate our rate of breeding. And that's still only really been applied very vigorously to only a very small handful of crops. And so we still have a lot of work to get breeding 3.0 out in the field and applied around the world. But breeding 4.0 is what I would consider where we're getting to this point where we're really designing genomes, where, of course, genome editing is the exciting new technology out there, but that really, we're going to make use of it, it requires that we actually have to have identified the causative nucleotides that matter. So it doesn't help if you just know that it's on this chromosomal arm if you're really going to start designing this. And so we also have to have enough knowledge of physiology to really make use of that knowledge. And so I'll be talking about, at the end, about how we have some routes to make into breeding 4.0. But I think breeding 4.0 is extremely exciting because we have an opportunity to move extremely rapidly with a lot of precision and do things in some very different ways. And so I think with those tools, the question is, what should we do with these incredible array of tools that we've built up over the last 10,000 years? And so the first question I think we need to ask is, what should we be breeding for? And what should those targets be? Should we just inherit the decisions that were made 10,000 years ago, or do we start asking some new questions on what we should be breeding for? So we've already talked about this in a larger context, but I want to highlight that there's some good news about us trying to feed the world. The first one is that global hunger has dropped by 38% over the last 25 years. And so that's an incredible accomplishment due to people all around the world working very hard to make that happen. But the problem is still 22% of the globe's population is hungry. And so that's billions of, a little over 1 billion people who are hungry every day. And so we need to do better than where we're at right now. On the positive side, Hans Rosen always highlight the fact that essentially more births are happening per year right now and will be declining over the coming decades. So we are in the century where we're going to have to deal with an increasing population because of better medication, longer longevity. The population will still increase 50%. But if we can get through this period at a time, we may be able to stabilize the globe's population. So with those two constraints, how do we address these issues? Well, the first thing is, could we put more land in production? And the answer there is pretty much no. Essentially, our crops and forages already occupy half of the habitable land out there, and essentially livestock with about 3/4 of it, and the crops for the other quarter of our agricultural land. And because of global climate change, it looks like actually productive land will likely decrease over the coming century, not increase. So we essentially have a fixed land, but what are we using that land for? Well, this is an example from the United States, but essentially most of our cropland in the US is being used to produce calories and protein. And essentially it's just 4 production systems, corn, soy, hay, and wheat, essentially occupy 88% of all that land, and almost all of those are being used either to produce calories or protein. So when we think about land use and feeding the planet, it's a very few production systems that are really dominating it, and they're all about calories and protein. You know, Fruits and vegetables obviously provide us a lot of our flavor and nutrition and everything else, but they only, in the case of the United States, they only occupy about 5% of all the agricultural land out there. There's certainly lots of challenges because of disease and transportation issues with them, but they're not a land use issue in order to produce those crops. And of course, we're facing issues like climate change, and a lot of the big challenges will really be about extreme weather events that our crops are going to have to deal with. And the question is, can we get those major production systems to be able to deal with these extreme climate events? And when people have started modeling these types of questions, for example, Hans Braun at CIMMYT, when he looks at the impacts of disease and climate change, we actually see global decreases in the total productivity in wheat and maize globally if we do nothing. But the green line is where we need to be if we want to feed this increasing planet. And I think the basic problem is the gaps between those red lines and the green lines are so massive that I don't think business as usual will be enough to make those come together. So I think the real question is, how do we close that gap? And we've already been talking about it. Giles and others have talked a lot about nitrogen. And I really do think a lot of it comes down to nitrogen. Nitrogen is the problem. How do we take this most abundant gas around us and make it into protein? And David Tillman recognized this problem over a decade ago in some of his nice work. He's highlighted that essentially in the next 45 years, we need to essentially produce more than doubling of the amount of protein for the planet. And so how do we get to doubling our protein output across the globe? And the problem is humans already dominate the nitrogen cycle. We already control 50% of the nitrogen flux every year. And just for comparison, we have a CO2 problem, but that's only being caused by us controlling 6% of the carbon flux in a given year. Essentially, 4 billion people on the planet are here already because of the Haber-Bosch reaction, and we're going to add another 3 to 4 billion people more. And so essentially, there is no more room with business as usual to produce more nitrogen and get more protein out. So we got to come up with some other approach than what we are right now. Now, I just want to remind people, how does nitrogen get to you? Well, in the case in the United States, we start out by drilling a hole in the ground. We extract methane out of that. We then create hydrogen off of that. We then combine it with atmospheric nitrogen in the Haber-Bosch reaction. We ship the fertilizer. We inject that fertilizer in the field. That we hope our plants uptake some of that fertilizer. They synthesize amino acids for it. They use it for photosynthesis. It's then remobilized. It then goes into our seeds. We harvest it. We then feed it to our livestock. We grow those animals. We slaughter the animals. And then we eat the animals. Agriculture has worked very hard on every one of those steps. And everybody's done a really good job of trying to make each one of their particular steps as efficient as possible. Obviously, the problem is the total number of steps across that process. And just to show you some of those inefficiencies, for example, on animal calorie conversion, it's quite inefficient, anywhere from 3% to 17% efficiency with things like eggs at the top. You look at the protein side, you'd hope it would be better. Not really. It's protein conversion efficiency from plants through the animals that we consume, only 3 to 31% efficiency, with eggs really being at the top as a very efficient system at 31% conversion efficiency. And our plants are not particularly land efficient in this process. So soybeans and maize are grown by the same farmers using the same equipment, sold seed from the same companies, all of them using the best agronomic practices that they can come up with to be as efficient as they possibly can. But maize yields 3 times what soybean does on those exact same acres. And part of the loss there is that soybeans are doing biological nitrogen fixation. It is not as it has an energetic cost to do biological nitrogen fixation. And you also look at maize's big problems. It's a very energy efficient crop. So in terms of energy in to energy out, in terms of megajoules, it's about, well, if you see it there, it's about a 10-fold increase in energy in versus energy out. So that's, makes a lot of sense why we use maize as a major crop globally, because of that 10-fold increase. But over half the energy that goes into growing the maize crop is coming from the fertilizer inputs. So if you could reduce the fertilizer inputs on that, maize would become even more efficient and start approaching what we're now using, biofuel grasses, or like miscanthus, for example, if we were able to reduce those fertilizer numbers. And also, if you look at the greenhouse gas emissions from a species like maize, almost 81% of its greenhouse gas emissions are coming directly from nitrogen. Whether it's from fertilizer production or from the nitrous oxide emissions coming off the ground from the lost nitrogen there. They're producing most of the problems with maize production, again, surround nitrogen. So I think that if you kind of summarize some of the problems, we're already using 1% of the globe's energy to fix the nitrogen. We have 50% losses when we stick it out in the field, and then we have anywhere from 70 to 97% losses as it goes through animal production. So I think we really need to ask the question, how can we do better than this? So obviously, some people have been advocating, and I think this is a useful thing to do, is try to change consumer preferences. And for those of us living in Canada or the United States or Europe, we eat very high, eat lots of meat, eat very high in the trophic level, and if we ate lower down on the food system, that would help. But we're a very small proportion of the total population. Most of the world's population are already eating these sources of protein. And almost all the increase in the globe's population are going to be people who are already, these are the only protein sources they have. So the change in consumer preferences in the big scheme of things are unlikely to have a really large effect in our ability to produce enough protein for the planet. I really think we need to go a different way. And I think we should look back at using specialization to do that. We started specializing our professions nearly 7,000 years ago. We are sitting in this room here with people with incredibly specialized skills. We are incredibly talented at a very small domain. Okay? Now the question is, can we take other elements of our system and make them work the same way? We're all working in agriculture, but essentially we are still using crops on almost all of our acreage that essentially were chosen 10,000 years ago, and are very generalized crops and animals in order to feed the planet. We have not specialized our production system the same way we've specialized our professions, which has made us become much more efficient at each of those areas. So I think what we really need to do is break up our agricultural production system, allow physics to do what physics is good at, let chemistry do what it's good at, and let the various elements of biology do what it's good at. And so just to remind everybody what our current production system is. Of course, we take fertilizer, we combine that with sun and light and water. We grow lots of different crops out in the field. They produce mostly protein and carbohydrates. We feed one stream off to livestock. Another stream goes to provide the grains to feed us. And then we take lots of those fruits and vegetables. We ship them long distances to provide a lot of the flavor and nutrition. So if we were to start reorganizing this type of agricultural production system into something that could meet those protein demands, how would we do that? I think the first thing we need to do is start thinking about where the energy is coming in for fertilizer production. And I think we have a lot of exciting opportunities because last year, wind and solar in the United States for the first time in an unsubsidized market became cheaper than fossil fuels were. And those prices are continuing to drop rapidly. And agriculture has some interesting needs for energy. A lot of it it could use a lot more off-peak power. So in integration with other types of energy needs, agriculture could be producing fertilizer and lots of other aspects if it could access off-peak power in an efficient way. And there were markets that really allowed one to tap off-peak power. And the way you can use electricity to fix nitrogen is essentially going back to old technologies. Before Haber-Bosch was invented, we were using electric arc to fix nitrogen, but we can do it a lot better than what we were doing 100 years ago, whether that's through plasmas or using fuel cells, for example, to split the hydrogen and put it into fuel cells. And John was just telling me about some additional approaches that could be used. But essentially, there's now ways we could use renewable energy to replace fossil fuels in that very first step of producing fertilizer. So the next issue is, how do we produce the protein? I think the key thing here is we need to get nitrogen out of the field and into fermenters. And so when I mean fermenter, we've already been doing this for over 50 years now, or nearly 50 years. We've been essentially fermenting free amino acids, for example, lysine, in order to supplement Maize's lack of lysine for livestock. So you take ammonia, you add glucose to it, and you give it to a bacteria, and you very efficiently produce lysine. The price point of lysine now is incredibly low. It pretty much matches what you can get out of a soybean in China. And can we do fermentation at scale? Absolutely. Bioethanol plants have highlighted that we can ferment at a massive scale. 210 ethanol plants in the case of the US produce nearly twice as many calories as all Americans consume in a given year. And could we use those proteins in a different route? Sorry. Already companies are starting to create proteins that have been tailored to livestock. One company is producing proteins for fish food in terms of methiogens, for fish food, and other companies are starting to produce protein supplements that can be fed to range animals. And then Silicon Valley and the Bay Area around San Francisco is starting to get filled up with a series of companies that are essentially starting to create proteins that are tailored to people, whether that's synthetic milk with cow proteins or leg hemoglobin that's now being expressed in yeast and then making a veggie burger that actually tastes like a burger. And I've had it, and I couldn't tell the difference between it and a regular hamburger. So, I think we now have the technology to make that process much more efficient. So how does that change things on the field side of agriculture? Well, we're taking the protein, the animation's not working, but essentially we're taking the protein out of the field, out of that system, and we're going to would require far less ammonia in the field. And of course, if we need if we're taking far less protein off the field in terms of our harvested grain, you don't need anywhere near as much fertilizer going onto the field. And that has tremendous environmental benefits, whether it's from reducing greenhouse emissions on the field or preventing dead zones of essentially our fertilizer that goes down waterways such as we have, killing off portions of the Gulf of Mexico. And then I think the final thing we need to look at is how can we make our fruits and vegetables more efficient and deal with some of the issues we're facing with climate change and pests. And here we already heard yesterday from some of the companies talking about greenhouses. And some of the interference issues are happening again on this. So I don't know, there's a radio frequency issue they were mentioning with the controllers. So we're having, in terms of greenhouses, they allow us to have a much more efficiency. They allow us to use excess CO2, reduce transport costs, and are using off-peak electricity and are allowing us to really reduce the environmental variation so that people can demand a higher market value for their crops. And just one example of how well that's working, the Netherlands, a very tiny country, is now the #2 global exporter in food. So, and that's all because of greenhouses using renewable energy from windmills and so on to make that happen. So I think the opportunities to make fruits and vegetables more efficient is certainly here. So how does this change how nitrogen gets to you? Rather than having that long route, it would allow us to shorten up that route so that nitrogen is getting to people in a much simpler way. So from a breeding point of view, as a geneticist and a breeder, how does this change what our targets would be? In the case of livestock, Some livestock obviously would not be competitive, but 3/4 of our agricultural land is still forage and as grazing land. So cows are still going to be out there. We need something to still go collect things. But you'd be then breeding cattle to work with a different mix of forages and feed. In terms of fruits and vegetables, you'd be breeding for a greenhouse, you're breeding to enhance flavor and nutrition, and you're starting to worry a lot less about disease and transport issues. Much of the last 50 years in breeding of fruits and vegetables has been focused pretty much on disease and transport issues, and we've neglected flavor and nutrition. So I think now is the opportunity where we can really engage consumers again by breeding for what they care about, which is flavor and nutrition. And then finally, in the grains and forages, we're talking about reducing input, reallocating nitrogen back to roots, focusing on temperature and water resilience, and focusing simply on calories for those. So what are some of the innovative products that could come out of systems like this? One would be low-protein maize. Maize right now actually produces 8% protein on a per hectare basis. That's about 800 kilograms of protein. Just to give you a feel, what does 800 kilograms of protein look like? That's 14 head of steer. Okay? That's a lot of protein that's coming off a cornfield. Now, if we reduce the protein content on that down to, say, 2%, we could then, at the end of each growing, we still need the protein in order to do photosynthesis and to be an efficient crop, but at the end of the growing season, we would remobilize the nitrogen back down to the roots. Both the nitrogen and phosphorus go back down into the roots, just as any perennial crop does, and it would be then available for the following year. You'd put it back into a nitrogen bank. In terms of efficient greenhouses, we can use those plants that are efficient at doing photosynthesis, for example, maize, and giving our greenhouse crops essentially a sugar IV so that they could grow very efficiently in greenhouses when light is limited. And our grazers, you would imagine a system where we have a lot of grazing forages that have very high starch and sugar levels, but then we would be feeding them synthetic amino acids in order to make them work well. So the question is, how close could we get to this maximal efficiency? So essentially, one person requires 57 grams of protein, and on a per year basis, essentially 21 million hectares of land is enough to synthesize the entire carbon backbone of everything necessary to feed the entire planet in terms of protein, and that's only 1.5% of all the global arable land. So we could go a great deal towards maximal efficiency if we started using fermentation and some of these other processes to produce the nitrogen through the system. Now I realize we're not going to go fully to this, but for the billion people who are already hungry and the 3 billion people who are coming, can we use these types of approaches to help make them make sure they're fed well and nutritiously? Okay, so I want to change gears here and talk about some of the technologies that we've been using to essentially accelerate breeding to go to, say, some of those last 3 or 4 targets. And I want to talk to you about how we're using some technologies to identify the nucleotides that are necessary for breeding. So as a Maize geneticist, I started out with this world. I've got 80 million genetic variants out there segregating across my breeding pools, and I need to figure out which 100,000 I should go manipulate and go modify and make it so that each variety is adapted to the needs I want. And so we've worked on all those areas in gray up at the top, but what I want to talk to you about today is how we're using machine learning models, many very similar models to what you all have been talking about in remote sensing, but applying that to the genome to figure out what nucleotides are most important that we can go edit through genomic selection or through genome editing. And so what we're doing is we're doing machine learning essentially across the entire central dogma. But what I'll be talking to you about today is just essentially the machine learning we're doing across understanding how chromatin, which is how DNA folds up, and how using machine learning to understand the expression of genes. And so why are we using machine learning? Well, some of the key aspects are it processes massive amounts of data, It generalizes the rules from one portion of the genome to the rest. It allows us to take the lessons we've learned on one reference genotype and apply it to the rest of the varieties out there. And then models can really be used to determine if any given genetic variant matters. And unlike some of the machine learning models that people are sometimes using in image processing, I'll highlight that really the machine learning models we're seeing in DNA, working with DNA, allow us to open them back up, and we are actually learning a lot of functional rules of how things work from machine learning. And so the models I'll be telling you about are essentially one class of models where we take in one sequence and we ask, is the DNA chromatin open? What transcription factor binds there? Is the gene a pseudogene? And what's the expression level that sequence produces? The other class are we take two homologous sequences to one another that have a few differences between the two, and we ask the question, is allele 1 or allele A or allele B more highly expressed? And three postdocs in my group have really been pioneering this whole area. That's Catherine Mejia, Hai Wang, and Jacob Washburn. So there's two basic approaches. The first one is essentially a k-mer or a language-based approach. And this is with natural language processing. So when you type in a query into Google, these are the type of things that it gets processed by. But we're taking DNA sequence, breaking it down into a bunch of words, or what we call k-mers, and then we're treating them either as a bag of words or essentially vectors of words. And I'll show you a little bit more about that in a second. And the second approach is we're using the convolutional neural nets, the exact same things we heard about yesterday that are being used to process images coming out of the fields, but we're using it in the case of DNA. And so in case of those natural language processing ones, we break this DNA sequence down into its component words, we count them up, and then we fit them into a regression model, and we make predictions with that. The vector-based approaches are slightly different, a little bit more complex, but essentially the same basic concept. And in terms of taking DNA sequence and fitting it into a convolutional neural net, we essentially take our DNA sequence and we make a black and white picture out of it by one-hot encoding each of the base pairs. And then we essentially run standard convolutional neural nets over it, and we relate it to a trait of interest that we're interested in. For example, we take that sequence and ask the question, can we predict its expression level? And so when we're testing out whether these models fit well or not, we're frequently using what's known as the ROC curve, or the receiver operating characteristic curve. And essentially, this is the ratio between the false positive rate and the true positive rate. And so the way we've set up our training examples here today, a random expectation, the AUC would be at 50%, and we go along that diagonal there. And if we had a perfect classifier, 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 machine learning models at these areas, we can get tricked up fairly easily. And this is where I think it's really important and necessary for biologists to think very hard on how we set up our training and our test data sets. And what we found is that evolution can actually be quite tricky and can help mess you up in this whole process. But we can also use evolution to very carefully set up our training and testing data sets so that we don't get tricked up in it. And the first way what we're doing is we're using evolution to inform our strategies here. And the one is we're using gene families. We train on one set of gene families while we hide whole and entirely other sets of gene families and make predictions across them. The second way is we use ortholog pairs, as highlighted in the bottom, to do the sets of training, and then we predict on other sets of ortholog pairs. And so this is essentially hiding various elements of the evolutionary signal out there so it doesn't key in on just memorizing gene trees in order to make predictions. And so we found that this has worked quite well in making highly robust predictions. So one high in the group has used this to try to predict pseudogenes and whether or not genes are highly expressed. He is essentially using the 1,500 base pairs of the UTR and the promoter sequence and the same on the terminator end of the sequence, run it through a convolutional neural net. And what he's trying to predict is essentially the maximal expression level of all the genes in the Maize genome. And for this case, he broke them down to 3 categories, either essentially not expressed, those that are moderately expressed, and those that are highly expressed. And using this type of model in CNN, he was actually able to come with an AUC over 90% when he was trying to predict especially off versus highly expressed. So just by looking at the surrounding sequence, we can predict these extremely well, whether or not a gene is on or off, using just that surrounding sequence. The question that Jacob has approached is which allele is more highly expressed. And for me as a quantitative geneticist, To move these things out, to deal with heterogeneous inputs and really generalize our learning. And so a lot of the work we've done in the past in our machine learning has been focused just in Maize or just across these small group of species that have nearly complete genomes. But what we're learning is that we still run into a lot of limitations because those species sometimes have very different physiologies, very different allocations of resources and things like that. So what we're starting to change our focus to is instead of learning across all the plant species out there, we're starting to focus, say, if we want to really know how to breed maize and sorghum and forage grasses in that group, we're going to start studying all 1,200 species that are highly related to maize and sorghum and learn from there. And across those group of species, we actually capture a total of about 1.5 billion years of evolutionary history. So we believe that that's the route to go to provide more and more information to train these machine learning models, is to saturate the evolutionary tree around the species that you're most interested in. And so we're then taking these predictions, we're evaluating these predictions based on field trials. So we have all sorts of predictions now on heterosis and fitness effects. We can test those against the field trials we're running, and then we're starting to interact with companies to start doing high-scale editing to evaluate and make these changes. And so I think we've gotten to the point where essentially we have the tools in hand where we can identify functional SNPs. Machine learning is providing us the tools to integrate them. And so really, and these tools can be applied to any species that you are interested in working on. And so the question is now, we go back to what do we apply them for? And so the take-homes I would argue is, what should we be breeding for? Well, let's start really specializing production, leverage electricity, and get nitrogen out of the field. And then learning for breeding, it's all about tapping evolution to provide vast amounts of data, using machine learning to really help us find functional variants. And then I'd be happy to, we do a lot of work on the logistics and engineering of deploying all these ideas is obviously very challenging, and that's what we spend a lot of our other time on. But obviously I think a lot of that has to do with collaboration and working together in order to make that robust. And I'll end here with the people who did a lot of the work and highlight the three people in the middle. Cinta Romay, Peter Bradbury, and Sarah Miller are group leaders with my group, and really help make sure everything works and all those collaborations with those 1,200 people around the globe work. So I'll be happy to take any questions if we have time.